Hello, good morning, doctors. Myself, Dr. Santosh. I will be your microbiology faculty in ADRPLEXUS. This is what we are going to do now. We are going to start with the first thing the first thing in your mind. There are a lot of empty pages. So I'm just scrolling it up. Okay, you can use it for writing other thing. So basically here, this is where we are going to start now. Enumerate the microbiological agents. These things you guys already know, just like a quick revision. Okay. So when you talk about microbe agents, what are the things coming in your mind? I know all of you must have already passed your microbiology long back. So if you guys remember, so when you talk about microbiological agents, you know that. Let's start from the smallest one. That is your viruses. And then comes your bacteria. And then a little bit bigger than bacteria is your fungi. And then the biggest one is your parasite. Right? Okay. So these are the things. Now, there are something like this independent microbial agents, just to brief you, independent microbial agents means the bacteria, the microbes which can survive independently. So, those are what? Those are most of your bacteria, most of your fungi and parasite. Everybody, these things, they can survive themselves also, except few exceptions. Dependent microbial agents are important. One is your virus. Dependent microbial agent means it depends on the host. It depends on the host, right? So a virus always needs a host. Not only viruses, if you remember, two important bacteria. What are they? Rickettsia, and then your chlamydia. They are dependent on the host. Even your Coxiella burnetii, that's okay, fine. Mainly you remember virus, Rickettsia, chlamydia, they are dependent microbial host, means they depend on the host for everything. Whereas your bacteria, fungi, parasite, they can independently survive also and then they infect us also, right? This you know. Now, next, look at this one. Microbial agents easily cultivable. So, can you guys tell me among all these four things, virus, bacteria, fungi, parasite, which is cultivable, which we can grow in a culture media? Very good. Bacteria, right? Bacteria, only bacteria. Most of you said only bacteria. Fungi, very good. Okay, these two. Let's stick to these two. It is either one is your bacteria and other one is your fungi. So we have culture media for bacteria and culture media for fungi. So we are going to discuss everything detailedly. Can you tell me for fungi? There's one question frequently, even recent exam INI-CET where they asked, what is the culture media for this fungi and what is the pH of that media? And can anybody tell you? Yes, I think Achasa was saying SDA, very good. Sabro, dextrose, agar. And what was that pH for that SDA? pH of sabro, dextrose, agar? It's easy. There's a clue there. I see the idea of this micro is we want to make micro so easy for you. So it will be more of what we will discuss. At the end of this fourth day, you must feel that micro is one of the top 5 easiest subject. Okay, so that will be guaranteed for sure. So we will just discuss. Anybody? You can't remember? Okay, no problem. So listen. So remember the sabrode is somewhat similar to number 5, right? 5. It looks like 5. So the pH of sabrodextrose agar is 5.6 or 5.5, 5.5 or 5.6. Both are right answers. Okay. That was a question asked one of the INI series. So I'm going to mark a Q. This will be revision in your mycology also. I'm just for we are going to talk all important things here. That's it. Okay. Now look at next question. The microbial agents that are difficult to cultivate. So which one you can't cultivate? There is no culture media at all. Is it virus, bacteria, parasite or fungi? Virus, okay. Jitu, Asha, all answered. Preeta also answered. Very good. Okay. So the viruses you can't cultivate. Not only virus, there are some bacteria which we can't cultivate. Can you tell me? At least I need 2 bacteria, very important. One is causing STD. Yes, very good. And Mycobacterium leprae. Yeah, Asha, very good. Okay. So you can't cultivate leprae. And you can't cultivate your Treponema pallidum. That is your syphilis causing thing, right? You can't. So can you tell me this Mycobacterium leprae? So we are going to talk this topic tomorrow in bacteriology. But if you guys tell me, yeah, many of you answered virus, very good. Can you tell me that how do we culture this Mycobacterium leprae? We don't have culture media. So how to, there is something, there's an animal model. Can anybody answer it? That was a question. That was a previous year FMG some question. Armadillo, very good, very good. Armadillo and food pad of mice, excellent, excellent. You guys are really, I think you guys have really practiced very well. I'm so glad. And students who don't know the answer, no need to worry at all. You, when end of the 4th day, you will know all the things. Okay. So this is just only to, warm up everything. So you will be, it will be easier in upcoming class also. So it is 9-banded. Someone said 6-banded. Nandini, it's not 6-banded. It is 9-banded. Okay. It's 9-banded armadillo. Okay. 9-banded armadillo and food pad of mice. Very good. Food pad of mice. Yeah. Entrepreneur problem we'll talk later. Okay. Now, next another important question, again frequently asked, is what microbial agents capable of spore production? So, which are the two things, microorganism that produces spores? That's okay, Nandini. I see you written 9. It's fine. Can you tell? Spore forming, which form the spores? Bacteria, fungi, virus among all? One is fungi. Very good, Jitu. Fungi produces spores, right? And what about another important thing? Yes, that is bacteria. You answered Clostridium. Very good. Bacteria. In bacteria, we are going to talk again. Two important bacteria are forming. Okay. One is Clostridium. Most of you have told Clostridium. Very good. It's right. One more thing you guys are forgetting. The anthrax. Who causes the anthrax? Bacillus. Bacillus anthracis. So, very good. So, Bacillus anthracis, that is Bacillus family, and Clostridium are the two spore forming. In fungi, you have variety of spores. We have arthrospore, conidiospore, chlamydospore. That and all we'll talk in that mycology only, which are only important things we're going to discuss. That is easy only. You don't have to worry. Okay. At least now you should know the Bacillus and Clostridium are spore bearers. Okay. The microbial agent that is not capable of producing spores means then the other two only. What are they? Your viruses and your parasites. Okay. This is not that important. So I want to stress here in this chart, only 3 things are important. Please remember the microbes which are dependent on the host. That is your virus, Rickettsia, chlamydia. One question can, whenever I'm marking Q, it is like the probability of getting question from there is high. Okay. And microbial agent easily cultivable, bacteria and fungi, especially SDS, abro, dextrose, agar, you're going to remember. Okay. Then this, how do you culture Mycobacterium leprae, 9-banded armadillo? Definitely expected question. Food pad of mice. Okay, food. I'm sorry, I wrote food. I'm sorry, someone corrected me. I didn't understand what you wrote. Okay, food pad of mice. Okay, and then armadillo. And here, spore forming. In spore forming, only 2 bacteria, your Bacillus and Clostridium, which we are going to discuss tomorrow. Okay, that's all. Now we are going to the next one. Microbial characters. So there are 2 things. Microbial. See, I think this non-pathogen is not a relevant word. You can cut it. Either there is commensal or pathogen. That's it. Okay, commensal. Commensal, and what happened, they nicely grow in a body. When the condition of a body changes or whatever, no, it just becomes a pathogen. So now please give me some example of commensals. Recently, I don't know, it's a FMG or in a NEET question, one question was asked. They told identify which of the bacteria is commensal was. It was a very easy question. Now you guys are going to tell me the example. Okay. So, you're going to tell me in skin, you're going to give me an example. In oral cavity, you're going to tell me some important commensal. And where in your GIT, you're going to tell some commensal. And in the female genital tract of female, you're going to give me an example. At least this much is more than enough. So, now tell me in skin, which is the commensal you're going to tell me. Staph aureus, Nandini, I don't know, is this for my question? Can you write it again, student? Doctors, in skin, which is the commensal? That was the question asked. Which bacteria you see in the skin? The name itself has clue for you. The skin, another name itself, there is a clue. Anyone, doctors? Yes, very good. Preeta has answered, right? Epidermidis, very good. Okay, so that is your Staphylococcus epidermidis. Epidermidis. So, okay, this is fine. Then what about the Staphylococcus aureus? Is it a commensal or is it a pathogen? So, I'm going to mark a Q here. So, don't forget Staphylococcus epidermidis is a commensal. Okay, commensal. It causes disease also, that we'll talk later. What about Staphylococcus aureus? Is it a commensal or a pathogen? Pathogen, very good. But please remember, this might be a question. Though epidermidis is the most common, Staphylococcus aureus, in 30% case, only 30% case, it is commensal too. It can be a commensal. Okay, commensal. That is one question. And if it's a commensal, which is the most common site, can you tell me in our body, in the skin, which is the most common site? Very good. Many of you answered pathogen. Very good. Nandini, Preeti and Asha. Very good. Can you tell me if it's a commensal, which is the most common site of this Staphylococcus aureus? And that's what we give a special ointment also for that, especially during surgery time to prevent this infection. We are giving an ointment. I want the answer for which is the common site for the Staphylococcus aureus. Yes, yeah, Ananya, very good. So someone asked what is a commensal pathogen? Yes, I'll tell you. So once again, before your answer comes, when a commensal means the bacteria, it lives in peace. It lives in peace with our body. Means it doesn't bring any harm. You know, it lives there. Either it gives us some benefit or maybe doesn't give benefit, but it doesn't cause any harm to us. That is a commensal. They are good people. Only when the condition provocates, immunity goes down, or when the flora thing changes, then you'll have a problem. Okay, that's the thing. Okay, I think Ananya understood, right? And pathogen means they are always pathogen. When this bacteria is there in your body, means they are dangerous, they are killers. Okay, that's pathogen. Okay, now tell me, so which is the common site of Staphylococcus aureus? Nares, very good, very good. And the topical mupirocin, very good, Nandini, right answer. So Staphylococcus aureus, the most common site is in the nasal, here in the nasal flares. Okay, anterior part. And then for this, that's what the treatment to kill this, use mupirocin ointment, right? Okay, very good. Topical, you give topical, very good. So Staphylococcus is mostly pathogen, only 30% case, fine. What about the oral cavity? Oral cavity, who's going to tell me? Oral cavity, we have a lot of commensals. What is that big group name? And if you remember, that is one of the most common cause of subacute infective endocarditis in a damaged heart patient. That's a question also. Which is that group of bacteria called? Staphylococcus viridans. Acha? Acha, is it Staphylococcus viridans or something else? Streptococcus viridans. Very good. Okay. Yeah, again, doctors, don't feel like if you don't know the answer and all, this all we are going to discuss. Now we are going like in a little bit advanced that, you know, you will get acquainted with it. Okay. When tomorrow everything will study, you will understand everything. Very good. So this is Streptococcus viridans. Okay. And you know, even Streptococcus viridans is one of the common cause of subacute infective endocarditis in damaged heart. Right? Because Staphylococcus epidermidis is the most common cause of subacute infective endocarditis in prosthetic valve, right? Prosthetic valve. Any patient having a prosthetic valve, if he is getting infective endocarditis, you will be telling what? Staphylococcus epidermidis. If someone is having a damaged heart, then it is viridans. Okay, right. Okay, good. Okay, now next. Tell me some example of the GIT. GIT, you have to tell me some important bacteria that are commensals. One is a huge group of anaerobes. In fact, they are the dominant in our gut flora. Lactobacilli. Jitu, wait. Jitu, it is not lactobacilli. Lactobacilli is right, but in some other place. Yes, Bacteroides. Yeah, Nandini has told, right. GIT, Bacteroides. Bacteroides, it's the anaerobe. And you, if you remember, anaerobe, this is also a question. This is the most common one, followed by Escherichia coli. Okay, followed by Escherichia coli. So for every 1000 Bacteroides, you have only one E. coli. Remember that. So it's a huge amount. That much amount of bacteroids we have. Okay, and this is an anaerobe. We'll talk about it. Okay, fine. Now in the genital tract. So this genital tract is the one someone answered. In genital tract, Jitu, Karan, you guys told lactobacillus, right? So that is common in your genital tract, vaginal tract. Okay, now one question for you guys. This also we're going to study later. When the lactobacillus goes down and some other bacilli goes up, there is a disease condition. When I say some other Gardnerella vaginalis bacteria and mobiluncus, when they go high, they cause which disease? Okay, Bhavadarni, very good. Achha, everyone answered. Bacterial vaginosis, very good. That is, we're going to discuss in detail. Clue cells, pH goes high, and then this thin discharge will be there. All those things when fishy odor, everything we're going to discuss it tomorrow. Okay, so basically that is bacterial vaginosis. That's all. Okay, good. Now these are our commensals. What about our pathogens? Pathogens, you know, anything we just told now. First one is your Staphylococcus aureus, we told pathogen. Apart from that, everything like Salmonella, Shigella, all important bacteria, most of the important bacteria in a body, they all are pathogens only. Okay, that's all. So this is the microbial character part we studied now. Okay, that's all. So please, here again, commensal, don't forget your Staphylococcus epidermidis and don't forget your Streptococcus viridans. Okay, and Bacteroides also, Lactobacillus also. Apart from that, see, there are few other things which I'm going to write it down. Okay, like diphtheroids, not diphtheria, diphtheroids. Diphtheroids is brother of diphtheria or sister of diphtheria, Corynebacterium diphtheria, which is totally harmless, useless. Okay, so that will not cause any infection. And also there are other few, you don't need to know all other extra things in it, but this is for important. This is for our exam purpose. Remember, these are the important commensals. That's all. Okay, right. Now let's go to our next topic, the scientist part. Okay, scientist part, which many people find it like, oh God, how to remember, it's too much and all. No, it's not really that much difficult. So I'll give you the shortcut how to remember. Okay, so we have Louis Pasteur and Robert Koch. Please remember, before going to writing this, you might not have this funny picture. You don't worry. I just want to only show this. See, remember, this is our Louis Pasteur, right? This is our Louis Pasteur. So what he's doing, he's given a car for a car wash. Okay, just remember the picture. I'll explain you what. And look at our Robert Koch. He's having a hanging, okay, stained cat is hanging down. This is the picture. Okay, clue. Now, how do you remember? You guys can, if you remember, totally fine. If for some students, doctors who find it that it's difficult, sir, I have so many subjects to remember, I can't remember this scientist and all. No? This is for them. I made small, easy things for you how to remember. Okay. So the shortcut is very simple. Louis Pasteur, you remember like this. Louis Pasteur, what he does, he gives, he gives his car, he gives his beautiful car for a liquid wash. Everybody needs to wash their car, right? So liquid wash. So, okay, this is a shortcut. Now everything you will get here. Okay, though the points, everything is mentioned here. So G, why did I put the word G here? What is that he invented? Doctors, any guesses? G for germ theory. Okay, the germ theory was given by our Louis Pasteur. First of all, he is the father of microbiology. It can be one question. Father of microbiology can be one question. Okay, number one. Okay, he invented the germ theory. It can be a question. Next one, V. Why did I say V? He invented the vaccine. Now you guys are going to tell me what are the 3 vaccines he invented. That's what I made this clue car. I made this clue car for 3 vaccines. What are the 3 vaccines invented? Cholera, very good. Cowpox, Preeti? No, it's not cowpox. Anthrax and rabies. Very good. You guys got it. Okay. See, the shortcut is C, no? So C for cholera, A for anthrax. Very good. And other vaccine is for rabies. He invented the 3 vaccines. Okay. That's what gives car. Okay. Right. Now, what about S for? S stands for? Who is going to tell me? S for? What did he invent? How do you clean all our surgical instruments, media? Very good. Sterilization. Okay. Very good. Bhavitran Nandri told. Okay. Sterilization. So he invented the sterilization. All your, the name itself says Pasteur, no? Pasteurization. That means it just says, no? All your techniques, pasteurization technique, your autoclave, even your hot air oven, most of the concept was given by him only. Okay. Any sterilization, you think about our Louis Pasteur uncle only. Okay. He is the main one. Okay, Louis Pasteur. So first one. Now next, what about this liquid? Why did I say liquid wash? Because we have 2 types of culture media. We have a liquid media and we have a solid media. We are going to discuss it later. So liquid media. The liquid media culture was invented by again our Louis Pasteur. Liquid media. Even if you forget also, L stands for Louis, L stands for liquid media. That's all. These are the things you have to remember. And of course, he did this fermentation principle also. He proposed fermentation. That was not much asked, but you can remember that also. Okay, got it? It's very simple. So Louis Pasteur gives his car for liquid wash. You remember, this is, you will never ever forget in your life. You will answer it easily. Okay, yes. Now let's go to a next person, Robert Koch. Robert Koch is like the next genius after Louis Pasteur. So he is basically our father of medical microbiology. Okay, or modern microbiology, whatever. Okay, he is the one. Now let's state how to remember this. So Robert Koch, he had, I showed you a stained cat was hanging, no? So he had a stained, Robert Koch is having a stained cat which is hanging from the solid roof. You saw the picture, right? You see this beautiful cat, it's hanging from a, everyone loves cat, right? So this cat is hanging from the roof. This is actually Robert Koch's cat, stained cat, okay? Stained cat hanging from a solid roof. Solid roof. No, roofs are usually solid. No, you can't hang from a liquid anything, right? Okay. So now let's see. Why did I say all these things here? Now you will understand why did I say. When I say stained, what did I stain? Because the staining technique. The staining methods were invented by Robert Koch. The simple stainings was invented by Robert Koch, number one. Now, who is going to tell me the three, cat means the three microbes he invented. What are the three microbes? What are three microbes, doctors? TB, very good. One, T stands for my TB, right. What about C? C, cholera, very good. So, yeah, Bhavadharani answered. Cholera, very good. What about A? Anthrax, very good. Okay. So, he invented these three important cat microbes. I simply call it as cat microbe. Okay. That's it. This is very, very important. Okay. Now, next. Hanging. What do you mean by hanging? What is that? To check the motility of a microbe, what do you do? What is that method called? Hanging drop method. Very good. Okay. I think Karan answered it, right? Karan answered. Very good. So it is a hanging drop method. So okay, these all are easy. Don't have to worry. Okay. Hanging drop for check the motility of the, we have many methods to check the motility. This is one of the method. Okay. Motility. To check the motility of bacteria. Right. Now, what about this? I told solid roof. That means which culture media invented? Liquid or solid culture media? Solid culture media. Very good. Okay. That's above the name. So that's all. Solid culture media. Okay. This is the main thing. Apart from that, this testing the disinfectant we are going to talk about also. Third disinfectant, how to test he invented. And of course, there's something called Koch's postulate also. I'll talk you briefly about this Koch's postulate. In Koch's postulate, okay, just write it in the side. Okay. Koch's postulates. It's very simple. When you read it, it looks like very hard. That 5 points of the Koch's postulate looks very hard, but it's not. It's very easy only. So what happened? Koch's postulate said that every microbe, I'm going to write microbe. Every microbe or bacteria, it causes a lesion. Okay, it causes a lesion or a disease. It causes a lesion or a disease. Some disease, let's say lung example. Some disease, it causes some lesion. Okay. Now, that lesion, this lesion whatever you got, from there, this lesion, you culture it. If you culture it, there will be growth. There will be growth of that organism. You will see the growth of the organism. You will see growth of the microbe in the culture, right? You will see. Okay. Now, from that growth of the microbe, now from this growth of the microbe, the culture, whatever you culture, the colony, usually we call it as colony. The growth of the culture is seen as a colony. See, colony. Now, from this colony, you pick the colony and you put into an animal, any animal model. You put into an animal, okay, this is animal. You put into animal model, then animal gets what? Animal also will get lesion in animal. You will get lesion in the animal. Now, from that animal lesion, if you are able to culture again, you should be able to isolate the bacteria. You should be able to isolate the organism. Very simple. You understand? First of all, a microbe, a lesion. From the lesion, you do a culture. Then from that culture, you bring the colony, you inject into an animal. That lesion should be in the animal, and from the animal again you are taking that from the lesion you're taking and you're putting into culture. The organism should be isolated. Very simple. Okay, they will not ask these things and all, but still you understand what the Koch's postulate means. Now the last point here is, which is the fifth point, that is important. This will be a question for you. What's the fifth one? Every bacteria, every microbes, what is involved for their pathogenicity or infection, which is there? They have something what? Gene. Every bacteria or any microbe, the gene is the reason for infection. They have a gene or genetic substance that is the reason for infection. Okay, that's all. This is the last one. This is the modified one. This is the modified Koch's postulate. Rest 4 were there already. Okay, This much only you have to remember. Rest all you remember. Okay, so don't forget staining, microbe, hanging drop, solid culture and everything. That's all. Okay, very, very simple. I'm sure that you guys will remember it. Okay, fine. Now let's go to our next one. Paul Ehrlich. Okay, this is very important. So who is Paul Ehrlich? He is father of what? Can anybody tell me? He is father of something. He's father of, is a father of chemotherapy or is a father of antiseptic surgery? You will get confused there. Father of chemotherapy. Very good. Okay. So see, it ends with CH also if you forgot also. Okay. He is father of chemotherapy first of all. Father of chemotherapy. Okay. That is fine. So what did he do? What did Paul Ehrlich did? You know what? He simply passed the SATS exam. There's a SATS, you know, SATS exam. Nowadays, these exams are popular for abroad. SATS exam. So for Paul Ehrlich, he cleared the SATS exam. So when I say SATS exam, what is this? Yes. Anything reminds you when I say what did he find? Why we call him a father of chemotherapy? What did he find? What was the drug name? That is also called magic bullet for syphilis. That was the first drug which was given for syphilis. Any guesses? Streptomycin somewhat, but ideally that was called salvarsan. Salvarsan. Sweta, you meant Savlon or it meant salvarsan? Okay, this is basically salvarsan. It's a sulfonamide compound only, salvarsan, which is also called magnetic bullet. Magnetic bullet to kill which one? Which bacteria? Your syphilis, first syphilis. That is, that time for syphilis they use this one. Okay, so S for salvarsan, number 1. Very good. What about 2? A. So A is, there's a stain. Okay, which stain is that? A stands for which staining method you know? For Mycobacterium tuberculosis, which is the stain you're using? Acid fast stain. Very good. Remember acid fast stain. This can be a question because remember, Robert Koch is the one who invented the tuberculosis. He invented the tuberculosis. That's what, you know, he's called, it's also called Koch's bacilli. But he didn't invent the acid fast stain, though he invented the stained cat, if you remember, stained cat, but he didn't invent the acid fast stain. So please remember, when they ask you acid fast stain, who invented? It's not your Robert Koch, it is your Paul Ehrlich, the father of chemotherapy. Okay, please remember that. Okay, acid fast stain. And T stands for toxin. Toxin means, I mean to say, toxin and antitoxin standardization. Toxin and antitoxin standardization. Okay, remember that. So what is the toxin antitoxin standardization? Can you give me some example? There were 2 important questions also frequently asked based on this toxin, especially in diphtheria. There is a toxin antitoxin. This he didn't find. This is an extra question. Okay, this is not related with Paul Ehrlich. I'm just asking you. Toxin antitoxin reaction, one reaction we used to check in diphtheria. What is the name? That is a question frequently asked in your exams. What precipitation test is that? Ehrlich gel. Very good. Very good. Who answered it? Yeah, Preeta and Nandini answered. Very good. Okay. So that test is called Ehrlich gel precipitation. You call it as Ehrlich gel precipitation. Right. Okay. Just one example. So same way. So standardizing a toxin antitoxin was invented by your Paul Ehrlich. Okay, fine. Now next one. Last year's stand for they found something. He found something, some chain. What chain it is of an antibody? It is the side chain. Okay, he found the side chain theory. He found the side chain theory of antibody. Very simple. So SATs. I know, guys, when you go to microbiology, you feel like you are totally out of your medicine, no? Because we are talking about a microbe. We are talking about the bacterial microbe, right? So that's what it looks like that. So anyway, the introduction part is, it'll be like the stories only. And when you go to the disease and all, now it will be more fun and you learn more things, okay? Important things. So, but these things are important for your exam. These were asked in your previous exams. So please remember, okay? So this is a Paul Ehrlich for SATs. That's all. We're done with this. Now, Joseph Lister. So Joseph Lister. So I hope it's clear, right, doctors? Whatever I taught, it's clear, right? It's understandable. Am I too fast or not? No, I hope it's not too bad. Yeah, okay. Now let's go to our Joseph Lister. So who is Joseph Lister? Thank you, guys. Thank you. I hope it's understandable. Now tell me, Joseph Lister. What is his father of? He is father of? Anybody? Any guesses? See, Listerine, you know, no, Listerine mouthwash is very antiseptic mouthwash. Listerine is antiseptic mouthwash, you know very well, very popularly used by all of us, right? So, what is that then? And so, that is basically he is father of antiseptic surgery. Remember that. You already have a clue there. Father of antiseptic surgery. Okay, this can be a question asked also. Recently, one of the exam it was asked. Father of antiseptic surgery. Okay, so remember that. Some medical office exam somewhere, this question was there. So, this is the only point you have to remember, nothing else. It's already, everything is given here. Listerine mouthwash. And yeah, he invented also, he invented that Listeria monocytogenes was pathogenic to humans. Okay, we'll talk about Listeria monocytogenes in bacteriology class. But okay, remember that. So, this is all Joseph Lister's participation. Okay, apart from this, we have some other important. Now you guys are going to tell me. I'm going to ask which one who invented. You guys are going to tell me. So let's start with the simple microscope. Who invented simple microscope? You have to tell me. And you have to tell me who invented the electron microscope. You're going to tell me. And you guys are going to tell me who invented the vaccine and who invented the PCR. And of course, penicillin, everyone knows. I shouldn't even ask this question, right? So tell me now. Who invented the simple microscope? Yes, Nandini. Anton von Leeuwenhoek. Very good. This is actually Anton von Leeuwenhoek. Anyone else is answering? Leeuwenhoek. Okay, Anton von Leeuwenhoek. Fine. Simple microscope. What about electron microscope? It starts with E only. It's not that difficult. It used to be asked in my time. This question was frequently asked. Now, yeah, Ernest. Very good, Nandini. Ernest Ruschka. Right. Ernest Ruschka. So, you already have a clue there. See, electron E for Ernest Ruschka. You don't even have to use your mind or, you know, revise and all. It's already there. Very simple. Penicillin, all of you know. Alexander Fleming. Okay. What about the vaccine? This question was asked. Okay. This was asked in recent one of your, I don't know, INI-CET or NEET. It was asked. This question was there. Or it's in MRB exam. I'm not sure. This question was asked. Now tell me. Who invented the vaccine? Which was that first vaccine? Yes, very good. Jenner, Edward Jenner, right? Edward Jenner. And which was the vaccine? Smallpox. Very good. Smallpox vaccine. Very good. Okay, nice. What about the PCR? PCR you can expect. PCR question been a while. Yes, it's Caribe mollis. Yeah, okay. It's fine, Pritya. You mean to be Caribe mollis. Very good. Right. That's all. Okay. Caribe mollis. That's all. These are the things for scientist point of view. These are the questions they can ask, okay, from the book or whatever. So, don't need to worry about. If these things you know, it's more than enough. You will easily get it. Okay. That's all. Now, let's go to a microscope topic. Microscope is again very, very important. You know that. Here in microscope, yeah. So, this is the microscope. So, we have what? We have two. Microscope, one important is the based on the light source, they are different, okay, and electron totally for most of the microbe, light is the source, but for electron microscope, electron is the source. That's the difference, okay, that's the difference. So when you talk about the light source, see the compound microscope is basically, it's called light microscope, the basic microscope which we are using in the lab, okay, light microscope. Now for everything, light is the source, but different type of lights are used. For light microscope, something we are using. For dark field, we are using something. For fluorescent, phase contrast is the same only, nothing much. Now, you guys are going to tell me which are the lights we are going to use. At least, you guys have to tell me for fluorescent microscope, which light we are using. Okay, Nandini, you said transmitted light. Okay, yes, right. For fluorescent, it is UV light. You already have a clue there. U is there, so UV light. We are going to discuss it in a while now. So, please remember this question was asked in your previous exam. UV light source for fluorescent light. What about light microscope? I think, who is going to tell? I think Nandini transmitter. It's right. Okay. See, light microscope, you have a T here. No, T. Light is T. So, T for transmitted light. See, everything in microbiology, everything you have clue there only. In your question only, you have the clue. All the things you have to do is little bit, you know, try to make this clues. It will be more easier instead of wasting your time in too much revision of micro, getting frustrated at the end moment. No, I don't want you guys to do that. When you're listening this class in this 4 days, your micro should be like one complete revision and you 70% it should be in your mind. Okay. Then by 1 or 2 revisions and solving questions, you will get it easily. Okay. T for transmitted light. Very good. So this is also can be a question. Dark field microscopy, who is going to tell me which light is the light source, but what type of light source for dark field microscopy? That was asked in one of the exam. Even I think in my exam only long back. In Ames exam. Reflected. Very good. Mira has answered. Yeah, very nice. Very good. So see, already you have a clue. Dark, there is R dark, right? So it is not refracted. There is no option. There was refracted. Okay, it's not refracted. It is reflected. Normal reflected light. That's it. Normal reflected light. These all questions. Okay. Transmitted UV in this thing. Phase contrast is normal light. Nothing to know. Electron microscope, you know the source of light is your electron, that is tungsten filament. You use the tungsten filament, and that tungsten filament was producing the electron. That electron was used as a source for this microscope. We'll talk about electron microscopy in detail now in a while. Now, please don't forget the light source. Okay, in microscope, first thing, the light source you should remember thoroughly. Reflected light, transmitted light, UV light. Transmitted for T, so light, it's for light microscope. That's compound microscope. And then reflected is for dark field. And UV light is for fluorescent. Fluorescent too, you'll never ever forget because you have a big U. Fluorescent U is there, so UV light. Okay, that you will not forget. Okay, very good. Now this is important. Next one, magnification. So whenever you take a microscope, what are important? The important for a microscope is magnification, resolution, and the contrast. These are the three things important for a microscope, right? Magnification is the most important. Everybody needs a magnification, first one, right? And then maybe I should move this slide a little bit. Magnification, resolution, and the contrast. Okay, contrast. Contrast is basically the stain which you are using is the contrast. The resolution power. Okay, fine. Now we'll talk about magnification, then resolution power. So you should know here now. So when you take a microscope, see, this is your beautiful microscope given here. Okay, see this picture. So you have something important. There are two types of lens, right? See the one you see in the upper part here, whatever you're seeing, that is called eye lens or eyepiece. Eye lens, okay, that is eye lens. The one which you're seeing here or the objective lens. We have different types of objective lens. If you remember objective lens, we have 100x. You would have seen in your labs, right? In your microbiology time, you have 100x, there is 40x we have, 10x we have, and 4x. That is objective lens. Okay, that is one point you have to remember. Now, what other things, you know, stand and everything. Okay, you all know about that, not much to note. So eye lens has 10x power. This has 10x power, important. And this is your stage. Now you guys are going to tell me where is the contrast, where is your condenser. Sorry, you have to tell me where is the condenser located. That was asked in one of your previous Ames, that is at that time it was Ames exam, it was asked. Where is the condenser located? Is it located above the stage or below the stage? That was the question. Below the stage. Very good, Acha. Very nice. Very nice. Okay. The condenser is below the stage. Please remember, below stages was a question asked. Condenser, the condenser used in the normal microscope is usually below the stage. That was a question. Don't forget. Okay. Rest all okay. These are adjustments, adjustment, fine adjustment, low adjustment, all those things. No one is worried about those things. You have to think about the eye lens which you're seeing through is a 10x power, and then the objective lens has 4 different type of lenses. 100x, 40x, 100x is the basically one which you're using oil. You remember? In your microbiology time, you used to see the grams in all the staining smears. You put a drop of oil on it on the slide and you used to see. That's exactly this one. Okay. So remember that. So that is oil is 100x 40. This much only I would stress in here. Now let's go to a resolution power. This was asked in one of the exams. I think not on the Ames, some state exam somewhere this question is asked. So eyepiece magnification, we have, let's 10x, 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 10x. Objective lens, how many types of objective lens we have? Just now I told you, we have the 100x which is the oil, and then we have 40x which is very good for your to see the motility, to see overall you to see the structures, parasites and all. No, it's very good. And we have 10x which for usually for motility we can check with 10x, and then of course your 4x. Now total magnification would be multiplication of these two, right? So when you multiply these two, how much you are getting? You are getting 1000x. This is the magnification here, 400x. And then you're getting 100x, and then you're getting 40x. So the maximum magnification you see in the 100x field, that is oil immersion field. If they forget to give you oil immersion field, don't worry, oil immersion is 100x. So with oil, everything goes maximum. Okay, so the maximum magnification you will be getting with the objective lens, which is having 100x. Okay, very simple, one concept only, it's not a magic thing, it's easy. So this one you remember. Okay, that's all. Now now we have to talk about the resolution power also. I told microscope, a good microscope depends on magnification, resolution and the contrast. Contrast is a stain, we'll talk stain in a while. Resolution, when you talk the resolution power, you guys must be knowing, or resolving power you call. You have to tell me for three important things power. One, for your eye, what is the resolution power? Then for the light microscope or the compound microscope, what is the power? And then for your electron microscope, what is the power? Who is going to tell me? See, whenever you say resolution power, you are able to distinguish 2 points. You are able to distinguish 2 points. I mean, I'm able to see 2 structures or 2 points exactly. That is the resolution. So what is the power of eye? What is our resolution power? It's all with a 2 only. The clue is it's 2. For eye, it is 0.2 mm. Okay, mm. And light microscope, it's 0.2 micrometer. Millimeter, micrometer. For electron microscope, it was 0.2 nanometer. This is a question asked in one of the previous exams. Okay, resolution power of your electron is very high because that is the magic microscope. That is the best microscope we ever have in our in a medicine, right? Electron microscope. So remember that. Okay, 0.2 mm, 0.2 micro and 0.2 nanometer. Nano is the big one. Nano electron microscope. That's all. Okay, this looks little bit mathematics. There is no mathematics in micro, only this plays in microscope only. We have little bit of this mathematics. That is very easy, I think. You will remember. I'm sure you will remember it. Okay, that's all. Now let's move to our individual microscopes. So the first microscope was a compound microscope, right? This is now types of microscope, compound microscope. Good. So can you tell me what is the use of a compound microscope? Is basically we call it as a light microscope. Don't get confused. They are same only. Light microscope. So now what is the use of a light microscope? Generally, I'm going to write it here. Use only for 2 use. One is to check the stained smear. You're going to check a slide which is stained, gram stain, Albert, everything you see. Second one, you can check the motility. You can check the motility of the bacteria. Motility of bacteria. That's all. 2 use only. No need, just for our knowledge only. No one is going to ask about light microscope, but it's basic. You know it. Now, let's go to dark field, which is very, very, very important. So, in dark field microscope, you guys have to, this one question, how to convert a light microscope into a dark microscope. There's only one structure we have to change to convert a light into a dark microscope. Who is going to tell me which is the structure? Is it your eye lens, or it's your objective lens, or it's your condenser, or it's your light source? You have to tell me. Yeah, Rana, you're asking about motility, the bacterial motility. In light microscope, you'll use someone, as Rana said, you can see when you put in 40x 10x, you can see the motility of the bacteria. You know, right? We have a lot of motility of bacteria: darting motility, tumbling motility, sliding motility, gliding motility, falling leaf motility, so many things. We're going to talk about it. Don't worry, we'll talk it soon. Okay, yeah, I better. Yes, condenser, very good. I think many have told, Jitu has told, Preeti, light source is not exact word, but condenser is the exact word, right? Exactly. So that was also one of the question, one of the INI-CET question, condenser. So what type of condenser you're using? You use dark condenser. That's all. This question was asked in one of my exams. This previously used to repeat it. Nowadays they're not asking much, but you can expect always. Only one thing you change. Everything is same. In compound microscope, instead of a normal condenser, you put a dark condenser. Then what happened? You will see exactly this. The background will go like this. Background will become this is the background. Background will be dark. It will be dark and you will see the bacteria like this. Okay. So now, which is the base layer given here? What is this bacteria you're seeing? The spiral one. What is this bacteria? We are going to discuss, but if you tell now, I'll be very happy. Very good. This is basically spirochetes. This is spirochetes. Now, what are the three spirochetes you know? These are all important questions also. 3 spirochete bacteria. Very good. One is your treponema. Let's start from the big and small. You remember BLT. BLT is Bachelor in Lab Technician students, right? Our students. So the technician students are very good with spirochetes. Remember like that. BLT. Okay, BLT. So B for, yeah, B recurrent is, yeah, but Nandini, what is the B stands for? B for, let's go in a broad term. It is Borrelia. Borrelia. Okay. L for your, who is going to tell me L? L for Leptospira. Very good. Leptospira. Spiral, no? Spira. And the T stands for Treponema. Yes, many told that. Treponema. Okay, so fine. So I'm seeing this curved one. If I'm seeing this curved one, what is that motility called? Tell me that one. That's important. In dark field microscopy, you'll not only see the spirochetes, but you'll see a motility also. That is an important question asked. That is what important here. Corkscrew. Very, very good. Corkscrew. Corkscrew motility. Okay, very good. Corkscrew. So, but can you tell me something? There's one more name also. There is, especially for Borrelia, the corkscrew motility is also called something else. Corkscrew is frequently asked. Definitely corkscrew is one thing, because see, if you forget also, the spirochetes, it looks like a screw only. You see, like a corkscrew. The one like screw which you're putting, the nail, no? The screw which you're putting, it looks like that only. So that is enough to tell. But even if you forget also, there, yeah, yes, for spirochetes, fine. But there is one special, no, Preeti, tumbling is your listeria. It's called, this very less they ask, it's called lashing motility. Lashing motility, okay? The question was not asked, but in the multiple option, one of the thing it was given, Borrelia has lashing motility. Okay, but that is right. It's mentioned. It's mentioned in the books also. So it's lashing motility is a fine. It's a right word. But overall, it's a corkscrew motility. Okay, that's all. Very good. So you're basically seeing your dark field microscopy is to see the spirochetes and motility. Okay, spirochetes and motility also. Other bacteria's motility also you can see. So not always only this one. You can see motility also. So both are right answer for dark field. That's all. Okay. Now, fluorescent microscope. Fluorescent microscope is a very important topic again. You have to really know all the fluorescent stain and whatever. So, when we talk about fluorescent microscope, two things are important. One is what? We told, which light we spoke? Which light we took? UV light. One is your UV light. And one more structure which is very, very important. What was that structure? You're seeing here. Yes, this is a, what mirror it's called? Here it's written light wave splitting mirror. Ideally, you call it as what? It's called what mirror? It's not asked in the exam, don't worry. But then you have to, there is a mirror is there. That's called dichromic mirror. Dichromic mirror. So when the UV light hits on this dichromic mirror, UV light when hit on the slide, see you can see UV light hits on this mirror and then the light reflected to the smear, which is containing what? The smear which is stained by, the mirror which is stained by what? Fluorescent stain. It is stained by some fluorescent dye or stain. Now you guys are going to tell me for three important stains. One for Mycobacterium tuberculosis, which is the stain we are using, and for Plasmodium. Plasmodium means your malaria. Plasmodium, which one we are using, especially for malaria, which is the fluorescent stain we are using. And one more thing is for your fungi. What is important thing we are using? You guys are going to tell me, because you will definitely have one question from this. Very important. Okay, very important. Now, what is for Mycobacterium TB? Auramine, rhodomine. Very good. Acha, very good. Okay, that is right. You say aura. M is there. See, auramine. M for Mycobacterium tuberculosis. Other one is rhodomine. So, Mycobacterium tuberculosis. Very simple. You don't have to use anything. Yes. And Nandini, you said acridine orange for malaria. Very good. So, for Plasmodium malaria, you are using acridine orange. Acridine orange. I'll tell you how to remember. So, acridine calcofluor. Yes, for fungi, it is the calcofluor white. Very good. Calcofluor white. Right. Now, how to remember, sir? In exam, I will forget. Okay, you will not forget. Why? Mycobacterium TB already you have auramine, rhodomine. MM is there. For Plasmodium malaria, what you do, see, whenever someone in your family gets malaria, what you will give them, when someone got malaria and admitted in the hospital, what you would give them? You don't give them bread or Horlicks. You give them what? Orange. Remember like that. You're going to give orange for your malaria patient. Okay. That's very easy to remember. You will never ever forget. So, malaria patient, you will give what? Orange. Okay. You got it? Orange. Now, fungi. Most of the fungi, if you see, it's mostly Candida, Crypto. If you come to our microbiology lab and if you see this fungi growth, they are white color only. They are white pasty color. So it is white. So calcofluor white. You don't have to use anything, your brain. White means fungi. Orange means for whom you'll give orange, you'll give to a malaria patient. Whoever your relatives, friends admitted with malaria, go and give them orange. Acridine orange. Very simple. Okay. Now you have one question also there. They haven't asked many times that question, but you can always expect a question from malaria. The people, microbiologists can go crazy to ask that question. What is the technique we use that acridine orange staining malaria? There is a name for the technique. What is that guy's name? In Hindi, what do you say for a fatty guy? Someone who is fatty, what they call them? Mota, yeah, right. Okay. Mota, whatever you wrote. Okay. So this technique which we call acridine orange we are using, it is called Kawamoto technique. Kawamoto technique. Okay. So M for malaria if you forgot also. Otherwise remember, Motu got malaria and you gave him orange. Very simple. Motu got malaria. Your friend Motu got malaria. And what did you give him? You give him a delicious orange to eat. That's all. Okay. You will never forget. I'm still surprised why they haven't, this Kawamoto technique hasn't asked for this many time. But please expect this question. You will have it. Okay. Like last time what happened, you remember, right? Vibrio parahemolyticus. They asked it for Vibrio parahemolyticus. You use the blood agar. No, blood agar means you increase the salt concentration. That is the blood culture. Vagatsumo agar. Vagatsumo agar was never asked. Nobody, they left it. Suddenly in INI-CET that question came in few years before. So like that, they like this fancy name. So if you now you will not forget Kawamoto technique is basically you're staining this Plasmodium parasite with the acridine orange. That's all. Okay, nothing to know. So this you will know. Now definitely one question from this part. Okay, fluorescent microscope. Careful, every point is important. UV light, dichromic mirror, and fluorescent stain. These three important fluorescent stain. Apart from here, you can see here. See in this our notes, fluorescent isothiocyanate is mentioned. This is one of the stain commonly used. Most commonly, see, if you remember, this is a fluorescent stain we have. We also use fluorescent for many purposes. Can you tell me one test where we use fluorescent in syphilis? In syphilis, who is going to answer me that question? We are using one place there. We are using fluorescent. I'm going to write it in the top, okay? In syphilis, for one test, we are using this fluorescent. And also in rabies, we are using fluorescent for one important test. Who is going to answer me? So we are going to finish all the fluorescent one. Very good. Nandini has told Hefty ABS. Yeah, very nice. That's right. So fluorescent treponemal antibody absorption test, Hefty ABS, one of the specific tests for your syphilis. Now tell me rabies. In rabies, that is the gold standard test. That's important question also. Which is the gold standard test for diagnosing the rabies? Something related with your fluorescent only. What fluorescent is that? We are detecting the antigen. So how do you detect that rabies antigen? What is that? It's called direct fluorescent antibody test. Okay, there also you have the word fluorescent. Okay, that is called direct. This is a question asked again. Direct fluorescent antibody test. To detect what? You're going to detect the antigen. Okay, these are the two places the word fluorescent will come apart from this. Okay, FDA ABS for syphilis repeated question. It's a specific test, and that is also first antibody to appear. And here direct fluorescent for rabies. That's all. Okay, very simple. This one you remember. You have to remember this important. Don't the negri bodies is a diagnostic form for rabies, but that is in the postmortem. In postmortem, negri body is very catalytic. You know that rabies. But in the live person, you're taking the nape of the neck. Say you'll take the smear and you'll do this fluorescent antibody testing to check the antigen in the nape. This is all what I'm telling is the question. We'll talk in virology. Don't worry. But even now, if you know, it'll be more easier there. You know, when talking. Okay. So this all things are coming under your fluorescent microscopy. The whole page is full with fluorescent. Yeah. So that means fluorescent is very important. Now fourth one is your phase contrast. In phase contrast, nothing to say here. Only thing you remember, one structure which we using is basically you're using a contrast plate. That's all. You're using a contrast plate. Okay, just a contrast plate we are using, that will create what? The phase difference. It will cause, when using this contrast plate, it causes some phase difference. It causes different phase difference, and then you can see all the internal structures very well. You will see all the internal structures of the bacteria very, very well. Okay, that's all. So that's about the phase contrast. Nothing much there. We haven't asked much question. One thing to remember, of course, you can check the motility of the bacteria also here. Motility is very well appreciated, plus the internal structures of the organism, internal structures, motility and internal structure. Okay, that's all. I haven't seen any question from phase contrast for so many years now. Okay, but please remember, phase contrast means just you're using a plate phase pair, then the light causes a diffraction in different phase, so you can see all the internal structures nicely. That's all. Okay, fine. Now next go to the next one, our electron microscope. Very, very important. You have to know. So which is the electron microscope? You can say everything, but which is the one thing that you can't see in any microscope, we can see in electron microscope of all the microbes we have? Tell me, can we see? Yes, virus. Very good. Virus cannot be seen in any microscope, but only in your electron microscope. Viruses. Okay, first is viruses. Second one, what are the structures you can see? Can we see the live structures also? Live organism also in electron microscope? Prions, Preeti, even I am not sure about prions. Even this is an interesting question you asked. I have to check it. I don't think so. I'm not sure about the prions. Okay, I mean to say, you will see only the live organism. Excuse me. You will see only the only negative with electron microscope is you will see only the killed organisms, not the live one. That means you can't see motility and etc. Not the live organism. Because imagine that electron is so powerful one, it will kill everything. Nothing can survive in that amount of that electron ka heat. No, you can't get it. So that's one thing. Okay, please remember. Okay, now electron microscope, every point now you're going to answer me. First of all, electron source. Then what type of lens we are using. Then what type of slide we are using. Then what type of the media, the electron source and media, you have to tell me which is the media, lens, slide, and you have to tell me about the fixative you are using. See, I am stressing only on electron microscope because this is very important. Any question can come and asked many times. Fixative and also the embedding media. Now you are going to tell me everything. So electron source is coming from what? Tungsten filament. Very good. Yeah, Preeti, you told, we just discussed before. Tungsten film. Everywhere it is the light source, but only for electron we are using a tungsten filament. Okay, very good. Now, what about the media? The whole media, see in the picture, you can see here this big wave what you're seeing here. Okay, what is that? That is basically it's a closed system, no? So what you call it as? It's a closed system, so it is a vacuum. Very good, Nandini. It's a vacuum. It is basically a vacuum because you can't let the electrons to escape out. So it is completely closed vacuum. Okay, that was also a question. What type of lens we are using? Everywhere we are using the normal lens, the objective lens, eye lens. But here we are using special lens because electrons are coming. So which type of lens it should be? Electron. So it should be an electromagnetic lens. Very simple. So the lens is basically electromagnetic lens. Electromagnetic lens. Very simple, right? Electron, everything about electron. What is the slide? Can we use a glass slide? Everywhere we are using glass slide. So what do you think about for electron? Electron is hitting. So what type of slide it should be? Anyone, doctors? Can you guess? I know electron microscope is difficult. You guys must have forgotten, but everything is a question. That's what I'm asking. Electron microscope slide. It is not electron microscope slide, Dr. Preeti. It is basically, you call it as metallic slide. Okay, the name I want is metallic slide. Every other microscope you're using a glass slide. Here you be using a metallic slide, and that's what we are using different fixative. What is the fixative using in other things and all? If you see, normally you come to microbiology, pathology, we use the wax. Just use the wax, a liquid, that paraffin thing substances to fix it. But here we are using some special fixative. What is that fixative? Something starts with G. After F comes G. A, B, C, D, E, F, G. So what is the G substance? So that G is a type of aldehyde, and that is also used for sterilization process. Now you guys have to answer me. I gave you the clue. Glutaraldehyde. Very good. Okay. So that is basically glutaraldehyde. Don't forget the fixative here is glutaraldehyde. Okay. And the same 2% glutaraldehyde. Here also it's 2% only. Was some 1.5 to 2, but it's number 2 only. It's also used where the SIDEX solution, which also used for, we are going to talk in a while. Glutaraldehyde used for sterilizing which articles? Doctors? Which articles? Which substances we sterilize with 2% glutaraldehyde? Yes, all the endoscopes. All your endoscopes. We'll talk about it in sterilization. You'll understand. Okay. Now, what is the embedding media? In embedding media, we will be using resins. We will be using resin substance. That's all. Okay. So this is electron microscope. Very simple and easy. Your microscope is over. You know, so it is one of the topic that you can't make any mistake. Okay. Yes. Now, okay, so we are clear, right? So very simple. We want to brief it again for the microscope part alone. You know very well the light source is important. So don't forget the transmitter light, reflector light, UV light and the uses. And don't forget this total magnification. Very easy. Just once you see, you'll know it. And the resolution power, I told you for eye, microscope and electron. Electron you have to remember. And compound microscope is otherwise light microscope. Everything, you know, condenser location is very important. Don't forget it. And also for dark field also condenser is important. You use a dark field condenser, dark condenser to convert it into a dark microscope. And examples, spirochetes frequently asked. We talked about it. Fluorescent microscope means that's very important. UV light. Another thing is different type of UV, the fluorescent stains which we are using. Okay. Acridine, auramine, rhodamine and your chalcofluor white. No doubt in it. You have to remember very well. Phase contrast is okay. And electron microscope, everything is important here. At least the slides you should know. And then this tungsten electron source and the fixative. You have to know. They are asked many times. This questions was asked in a different way. Okay. That's all. Now we are going to the lab diagnostic part. Generally, in lab diagnosis, what are the significance, role of laboratory in infectious diseases? Okay. Infectious disease, what are the things we have? So we we do lab diagnosis for three things. One is for diagnostic, mainly it's for diagnosis. Other one is for prognosis and follow-up. They are almost having same meaning only. Now, you guys are going to tell me, wait a second, yeah, it's coming. Now you tell me, diagnostic is for most of the diseases. I'm not going to talk about follow-up cases. Which are some diseases where follow-up is very, very important? Follow-up means to see the prognosis, whether it's decreasing or not. Can you give me at least two important diseases? One is tuberculosis, fine. Okay, yes, I accept it, but I expected something else also. One more thing. Okay, for TB, what you do? You will do the sputum smear. Again, you will do sputum smear and see the load has decreased or not, right? And anything syphilis, very good. That's what I was expecting. One important thing where prognosis we use is syphilis. And here, so here for TB, it is your acid fast smear you're checking for the prognosis. For syphilis, what you are using? Which is the test you're using? Myra, you have still syphilis, but tell me, Mira, right? It's Mira, huh? Tell me, which is the yes, VDRL, only VDRL. Only VDRL. We are going to talk about this tomorrow, don't we? RPR also, right? Okay. Prognosis. For prognosis, this we are talking about not the diagnosis, for prognosis. Prognosis of TB, you'll do acid fast and you'll check the smear decreased or not, number of count of the acid fast but decreased. For syphilis, the VDRL, RPR you're checking. Very good. That's okay. Nice. And then what about for HIV? HIV also important, no? You tell the HIV patient to come back and patient come back. What is that? To check the load, the treatment is working or not. What is the thing you will do? If I give you option CD4 count, RT-PCR, what are you going to choose? Excellent. Very good, Karan. So no more CD4 days. CD4 days are gone now. Okay. Now for prognosis, you are going to use PCR only. RT-PCR. Okay. RT-PCR. To see the prognosis, your quantification of the number of copies of this. Okay. So these are the three important. Of course, you do many diseases, not only this, even for your hepatitis and many things you do. But at least for prognostic value, these are very, very important. Microbiological point of view, these are very, very important. Okay. That's all. So this is the main thing. And follow-up is same follow-up. Most of the disease comes here. So this is okay. This is not, I'm going to stress. Now let's go to diagnostic part. Now, so this is basically they've given very nicely for which one. So when you talk about the microbiology, these are the tests available with us. One is your microscopy. Number one, we start everything with microscopy. And then we do culture and biochemicals, it should be culture plus biochemicals. Then the serology part, we'll talk about, and the molecular means your PCR methods, different type of PCRs. So here they given briefly. See, microscopy, we can use a direct microscopy or stain microscopy. Stain means the stain smear. When say direct microscopy, it means the wet mount. They're talking about the wet mount or hanging drop methods. Okay, not that. This is the briefing. There's nothing questions are going to come from here. It is to get an idea what is important. And serology, when you talk about serology, these are when you talk serology, one thing you have to come in your mind is ELISA test. Don't forget ELISA test and the immunochromatographic test, ICT, and we have many tests, but these are commonly used to test. Okay, ELISA and these two. Then, of course, we have culture, pathogen identification, antibiotic sensitivity, etc. Then RT-PCR. Okay, we are doing this only. Okay, fine. Now, individual. If you talk individual, for bacteria and fungi, this is right. We are talking about culture and microscopy. Please remember, that's important. For bacteria and fungi, always you will be starting with what? You will do when in microscopy, you always start with the gram stain, and then you do the culture, different type of culture medias we have, and accordingly we classify and we do it. Okay, grams and culture, that's okay. That is generally for bacteria. For virus, you don't have culture, that we already spoke. Virus, there is no culture. You don't have any culture. You directly go for serology only. That means you'll do ELISA, anything, and then you'll go for PCR. So this is serology plus PCR for any microbe, right? Even you remember for COVID time, what we were doing? We were not doing any culture. We were doing only serology, ELISA or PCR. PCR is the best confirmatory and everything. But if you don't have that much, then you go with ELISA. In ELISA, you can detect everything. It's your swine flu or it's your herpes or anything, serology or PCR, whatever. Parasites, mostly microscopy is enough, to see the eggs, because when we talk parasite, we're talking about the eggs, the worms, eggs, cyst, which all we are going to talk day after tomorrow. That's all. This is the briefing of in a lab diagnosis, what needed for bacteria and fungi and virus and parasite. That's all. These are the things, basic things that you have to remember. Okay, that's all. Now we are going to the culture methods. Very important place we have come is the bacterial culture. First culture method, and then we'll go to the culture medias. Okay, first when you talk about the bacterial culture methods, the first culture is called streak culture. So the question here they can ask is streak culture use of the streak culture. When you do a streak culture, you see this is a streak culture. That means you took a plate. I took let's say this is a blood agar plate, and then I got a sample, any sample. Let's just sputum sample. And what I'm doing, I'm just streaking it like this. See, I'm streaking this and I'm putting it inside the incubator. I'm leaving it in the incubator, overnight incubator, usually for 24 to 48 hours. And then next day I'm looking for this beautiful colonies. What type of colonies I need? Look at this, isolated colonies. My purpose is to isolate the colony. That is for streak culture. So streak culture is to get the isolated colony. Only when I get isolated colony, I can do biochemical test and I can do antibiotic susceptibility testing. And I can give the diagnosis of the bacteria, which bacteria it is. Right? Very simple. So for this, I need to do streak culture. Remember, when you streak it, you get isolated, separate isolated beautiful colonies. So can I ask a question? If this picture was given to you, this blood agar plate is given to you, I can see, can you see some pigment? Can you see that golden pigment there? Which bacteria is this? You had this question before once, I remember, in long back. It's a Staphylococcus aureus. Very good, very good. Everyone answered. Aureus, only in aureus, because aureus means golden pigment, right? So this is Staphylococcus aureus bacteria. We'll talk about it, don't worry. But if you remember now, it's very good. Okay. So this what? So I have diagnosed this bacteria very well. So streak culture is to isolate the bacterial colony and further proceeding. Streak culture. Okay, right. This. Lawn culture is a very important question. They ask also. So lawn culture is basically you take a swab. No streaking business here. You take a swab. You take a beautiful swab. Okay, you take a swab, and then you dip inside the organism you're seeing. Take a culture plate, and then you just swab it like completely, like a lawn, like a lawn. Like a lawn. You put it. Okay. After putting the lawn, you see lot of discs put it over here. So this is basically for what? AST, right? AST, right? Okay, someone did AST. This is for AST. So the lawn streak culture, yes. Once again, streak culture we don't do for putting the discs. That's only for isolating colony. Lawn culture, you're doing the color. You're beautiful. Let's say your house and outside the house you have a beautiful lawn, right? So lawn is even. No, all the grasses are even. Then only I can see the thing. So what you're doing basically, this is for this test. What test is this? Kirby-Boyer disc method or antibiotic susceptibility testing. We are going to talk about it in a while now fully. So Kirby-Boyer disc method, this is called, this is a question asked also, Kirby-Boyer disc method. So now what I am doing, you see, I put it the all the these are the drugs. Maybe this is an erythromycin, this is clindamycin, maybe this is a penicillin, maybe this is a clotrimoxazole, maybe this is a tetracycline. I put at the culture plates, okay, and then I'm seeing the zone, whatever you're seeing, no, this is called the zone of inhibition measurement. And then accordingly, I will say the drug is resistant or sensitive or intermediate. Then I'll give the report to the patient. Okay, that's the thing. Antimicrobial susceptibility testing. Yes. Any doubt here? Yeah. Okay. You're asking what is AST? Oh, sorry. Right. That's antimicrobial susceptibility testing. Okay, fine. Okay. We usually shortly we call this AST. Okay. No problem. Good. Okay. I think yeah. Okay. Preeti has answered. Good. Okay. Now. So now one question is there. Which is the media we are used to do the antimicrobial susceptibility testing? It was one FMG exam. This question. So nowadays FMG NEET means all mixing only, you know. They can ask you. So which is the culture media? What I put at this? Which media is this? Which plate is this? Ah, sorry, Preeti, it's not nutrient agar. We have special media. It starts with M. Muller-Hinton. Very good. Okay. So you use the Muller-Hinton agar. Please don't forget Muller-Hinton agar means for Kirby-Boyer disc diffusion method. Any related to drugs, any AST antimicrobial susceptibility testing, you will be using. Yes, Preeti answered, Nandini answered. Very good. Okay. Muller-Hinton agar. Okay. The students you forgot, please remember now. Muller is for drug. Mr. Muller, he likes to do AST testing. Okay, that's a Kirby-Boyer, Muller, they are together. Okay, Kirby and Muller-Hinton. By any means you have to remember. Okay, this one. Now a recent question asked, now because we are talking about AST, now we are going to tell about the see, I'm going to write it here so that it will be easy. Now what are other methods you know for the AST testing? I'm going to write it here once again. AST means is antimicrobial susceptibility testing. Now I'm going to write here AST testing methods. Antimicrobial susceptibility testing methods. One is number one, Kirby-Boyer disc method, disc diffusion, which we call it as disc diffusion. Fine. Which other methods you know? There is something called minimal inhibition concentration. Okay, once again, I'll write the full name. Okay, this might you might have forgotten, that doesn't matter. There is something called MIC. MIC is basically minimum inhibitory concentration. So there we have some methods called, like you put a strip, usually called E-test method, or dilution method, we call dilution method. I'll tell you how to do it. Okay, this is one method, MIC method, Kirby-Boyer disc method. Nowadays we can also do what? PCR. We can do PCR also. MALDI-TOF. These are automatic method. MALDI-TOF. These are the few test methods you can use for testing the antibiotic susceptibility, antibiotic resistance, or it is susceptible, intermediate. You do that test. Okay, let me see if we have a picture here. Yes, we have other pictures. Yes, I'll show you. We have it. Okay, no problem. I'll get in a while there. Okay, fine. So, okay. So, please remember all these are right answers. But in exception, I think in option they have given CAMP test. Where is the CAMP test is used? CAMP test, anybody going to tell me in that question previous? CAMP test is used for group B streptococci. Very good, Nandini. Okay, so this is no link with the antibiotic susceptibility testing. For antibiotic susceptibility testing, you use this disc method Or this E-strip method. E-strip method, basically it looks like this. You put a strip like this, you will get a zone like an elliptical zone. Okay, I'll show you in other pictures, okay, in coming classes. Okay, you remember now. And we have that one, and then PCR method, Malditoff, that's all. So CAMP test is for grouping, that's fine. Okay, fifth one is Malditoff. Prairie, it is Malditoff. Okay, I'll write it again for you. This is basically called M-A-L-D-I, Malditoff. Malditoff. This is an automatic method. You don't have to do, just you put it, automatically it will come. This is basically called matrix assisted. You don't need to all the name. It's a matrix assisted iron desorption assertion. You will get confused. Don't go that much detail. Malditoff is only, usually it's called Malditoff. Okay. It's basically a matrix assisted iron based method. Okay. Time of flight you call. You don't need to all the name. Just remember this only. Okay. That's all. Now let's go to the next one. So antibody testing method I've told you, other methods we spoke now. Now we are going to our next one, that is a stab culture. Stab culture is basically what you're stabbing someone. You're just stabbing with someone. You see, this is the culture plate. This is the tube. This is, let's say this is a tube. This is I'm having a semi-solid agar, some semi-solid agar. So basically when I say semi-solid agar, this is some motility media. Okay, motility media. Let's say it is a motility media. Now what I did, I'm picking my colony and see, I've inserted inside. I'm stabbing it with the wire. Okay, I'm stabbing the colony with, I have picked a colony in this loop and then I'm stabbing it inside. See, it is stabbed inside like this. I stabbed. And next day, you see, did you see this? The colony has spreaded like this. From center point, it has spreaded nicely on the outside like this. So what is the meaning of that? It's a motility media. So what is the meaning of that? So my bacteria is motile. It has just moved. Okay. So stab culture is used to check the motility and also for biochemical tests. A lot of biochemical tests also we do stab culture. Okay. So if you forgot, remember biochemicals and see, you already have your clue. B for biochemicals, stab biochemicals. Okay, biochemical, and for motility media also we can use this type of method for culturing. Easy. Okay, just only 2 points. So we have streak with the loop, and then you do the swab with the cotton swab, and then stab with another wire. So these are the types. Okay, types of the culture. Now there is one more method also very important. Pour plate method. Okay, pour plate method means you can have one, though this is bit obsolete nowadays, we're not using so much, but yes. Okay, so in pour plate method, what you do, in olden days, what happened, especially we do now also, but now all automated came. So to check the colony count, the main thing, this pour plate method, otherwise called also Miles-Misra. Okay, that's also important. Miles-Misra. Basically, the idea of this guy is to count the number of colony. Count the colony number. Can you tell me for which infection or which sample the count of colony is important? You have sputum sample, you have CSF sample, you have tracheal sample, you have urine sample. So many samples you are sending to us. For which one the count is important? Typhoid ka Preeti in a serological way. Yes, yeah, Mira's right. Mira right. Yes, Asha right. Yeah, urine. It's urine. Nandini, it's, yeah, Nandina's right. Karan, it's not CSF. CSF may any bacteria, it's infection only. You don't have to worry. But in urine, what happens, especially in urine, because we have lot of contaminants also mixed, no? Contaminant mixed. So we don't know it is a infective endocarditis also, Mohanram, not that much. We don't use it. Yes, for some of the tracheal samples we can use. Tracheal sample we are using some of the, if it's coming from deep bronchoalveolar, lavaginal we do. But ideally it's urine. You don't mix anything, urine. Because urine easily, see, rest all other fluids are sterile, no? So in any sterile fluid, you see one bacteria also, it is pathogen. It doesn't matter. You see a bacteria in CSF, it's pathogen. In blood, it's pathogen, sepsis. But in urine, you don't know, na? Sometimes it can be commensal. Example, pregnant ladies, they diabetic patients, they always carry a lot of bacteria, which is not causing any infection. So that is what, when you give the urine count, the clinicians, you people will coordinate. You will see and see if it's more than 10 to the power 5, it's a significant, so you give the drugs. So if it is less than 10 to the power 3 or 4, then you have to see patient is asymptomatic, then you don't treat. So for urine, so the idea is counting the colony is very, very important for urine samples. So that's what this Miles-Misra technique also came. Okay. So now what he did, first this one, he took the original inoculum, the colony, the original bacterial colony, picked from the plate. He took it, he mixed it very well. He mixed it very well with the broth. He mixed it very well. And then what happened? You see, he started diluting it in the normal saline or whatever. Everywhere you see it is taken here, it's become 1:10, then become 1:100, then 1:1000, then 1:10,000, 1:to this much. Okay, this is the range. Now from every tube you will take 1 ml and put separate plates. This is all separate plate. For this one 1:10, you are putting this first plate, then second plate, third plate, fourth plate, fifth plate. Now you will have this picture in exam. If you never know, they can give this picture and tell identify in which plate the colony count is taken. So you see, I can't take the first plate because the colonies are not clear. I'm not going to take the first plate because it's still confluent. You see the growth? I don't see any single isolated colony. I'm not seeing, no. Everything is together mixed. Here also it's mixed. I'm not going to choose this colony. And next one, I can, but you see still some places are mixed. I can't. I'm going to say no. Then what I'm going to go to the next plate. This is the first plate where I can see all isolated colonies. Now I'm going to take this as my range. There is a calculation for that is not needed, but if you say they ask you option A, B, C, D, and they'll tell you which of the following plate you will take it as the colony count. So then this plate number 4, I will take it as a colony count because I see the isolated colony. I see the isolated colony, not this one. This one very less. Here, which plate isolated colony is in, that plate is the positive, and that would be your number of colonies. Okay, right. Then UTA will talk about significant urobacteria. Now you remember the Miles-Misra. Okay, we call Miles-Misra, Miles-Misra. You can say whatever. So it's basically colony counting. Mr. Miles-Misra, he likes to count. That's it. Miles-Misra means counting. Everything he does is counting. That's all. Okay. So very simple. Don't worry. This was been a while. They never asked. If they ask, or if I have to ask a question from here, I would simply ask Miles-Misra is dash method, and I will write pore plate or streak or whatever I'll say. So you, by knowing pore plate, you can easily say this is a Miles-Misra. So Miles likes counting. Miles-Misra, he likes to pour and he likes to count. Just two things. He likes to do only two things. He likes to pour and then he likes to count. That's it. Pour and count. That is Mr. Miles-Misra's work. Okay. Rarely ask this question, but yeah, we will be ready for everything. No, you guys, it will be very useful. Not much to do. Okay. Now let's go to the culture media. It's very, very important topic because your whole bacteria depends on the culture growth only. So the main culture we'll do study tomorrow. Now the basic culture media. You have a lot of questions. The culture media. Now tell me, any culture, what are the basic constituents? What are the basic constituents? When I have a give you culture plate, if I tell, Dr. Saab, isme kya kya hai batao. I mean, I'm sorry, I shouldn't have Hindi. If I say you, sorry for that Hindi practice, it's coming. So, if I give you culture plate and tell you, Dr. Saab, please tell me what are the culture contents are there in a media. What are the things commonly you see? You will see a peptone. And the next one is your agar. That's it. Now, important question comes from the agar. You will have a question. Okay. Now, tell me, when you talk about the agar, yes, what are the concentration we are using? Which is the most common concentration? So, I'm going to do like this, divide the agar into like this, 0.2%. This question was asked in your recent exam, and 6%. You have to tell me which is the most common percentage of agar I'm using in a culture plate. Remember from a microbiology time, you must have studied in the practical, they must have taught you in your college. Otherwise also no problem. If you forgot, it's no problem, but make guess. Always, for any culture, for normal culture media, the percentage of agar we are using is 2, 2, 2. Okay, 2, 2, 2. Okay, this is the most commonly we using culture. All the culture plate, blood agar, your McConkey, whatever agar you're using, it's 2%. Which is the condition we use 0.2%? You're decreasing the concentration means what is the meaning? The culture becomes very loose. So when loose means the bacteria can move. So for motility, to check the motility, you're doing 0.2%. Now, very important point, 6% was a question asked. 6% agar is used to inhibit one important motility. Which is that motility and which is the bacteria causing that motility? It was asked in one of the recent exams. I don't know, it is the one of the medical officer exam somewhere, but that exam it was there. So many exams, I couldn't tell which exam they asked. Who is going to answer me? Which motility when you increase the concentration to 6%? Guys, you make mistake, no problem. I would say my students, no, answer something and make mistake, it will come. Okay, Karan said tumbling. No, Karan, it's not tumbling. Okay, I'll give another clue. This, yes, very good. Jitu has told it is swarming. Very good, swarming. Right. Okay. So basically, it is to inhibit the swarming motility. Now, question comes: who caused the swarming? Which bacteria cause the swarming? There are 3, 4 bacteria, but most common bacteria is Proteus. Very good. Okay, very good. Yes, Acha said, Preeta still. Very good. Okay. So now swarming motility, what you do in 6% concentration is basically inhibits the swarming motility. Inhibits the swarming motility of which bacteria? 3 girls will come in swarming motility. See, for if it's a FMG junior exam, Proteus is enough. But now we're going to NEET-PG and INI-CET, you should know all the motilities better. So how you remember? You remember 3 girls for swarming, though we'll talk later. 3 girls, they are having what party? Tea party. 3 girls are having tea party. Who are the 3 girls? I'll show the picture in tomorrow's class for a minute. 3 girls, 1 girl is Prochita. All my friends only, medical friends only. One is Prochita. Other girl name is Sarah, and there's one more girl called, her name is V. Pauru, not our Pauru parvovirus, this is V. Pauru. Okay, so now you guys are going to tell me. One is Prochita, Proteus, you told. Very good, excellent. Frequently asked question. Why did I say Sarah? Which bacteria is that? Serratia. Very good, excellent. You never know. This is the only red pigment producing bacteria they can ask. And Vibrio parahemolyticus. Excellent, Acha. I thought you guys will not answer it, but you told, because this question was asked in our INI-CET recent exam. It was asked. That's what I'm saying. Vagatsumo agar, no, when using the seafood, Vagatsumo agar, you have to grow it in a high concentrated blood agar. That's what. So it's fine. Para hemolyticus, eating the seafood. Okay, Proteus, Serratia, Vibrio. Why did I say tea party? Where is the tea word comes? Tetani. Very good. Excellent. Okay. Clostridium tetani. Remember these 4 people. I would not stress you in other like other motility. In this I will stress. In swarming me, you have to know this 3 girls, 1 tea party. Okay. 3 girls, they having 1 tea party. Okay. Prochida, Sara, Viparu and our they having tea party. You will never ever forget. So inhibiting the swarming motility, one is 6% agar. Not only that, they have the option it was given. What are the things you can decrease? When you increase the agar 1, and when you're adding charcoal, or when you're increasing the bile concentration, these are the things they can inhibit the swarming motility. Okay, because in culture plate, no, when they grow like this, they usually swarm. If you put like this Proteus is very like this, it grows like this is swarming. So it really disturbs other bacteria to grow. So to prevent that only we're using. And one more thing also you have to tell, which is that one special culture media used for urine that inhibits the Proteus swarming motility. Usually we use McConkey agar, but only for this case in urine to prevent the Proteus swarming, we're using that media. If someone is answering, that means then you guys are very good in micro. If you don't answer, no need to worry, because that is definitely something which you will forget. I know that usually even we also forget, but that's important media. For that is the best media for urine sample. When someone want to test the urine culture, UTI patient having urine, and if you want best culture plate, we recommend that only, not the McConkey, the other one. What is that? Something starting with C. It is basically clad media. Chocolate agar, Prita? No, Prita. It's a chocolate agar. Chocolate agar is a type of blood agar, no? So it is called clad agar, clad media. Cystose, lactose, electrolyte deficient media. We'll talk about it in urine, don't worry. But please remember clad. Okay, I'll write it here. It's basically cysteine, lactose, because it differentiates whether it's lactose or lactose, LAC, lactose, electrolyte deficient. Because of electrolyte deficient only, there is no swarming motility. Got it? So remember. In McConkey, there is swarming. It will grow. It's not every time, but you still, to some extent, it will not grow, but it will grow. In blood agar, to swarming will come like anything. So we microbiologists will say the clinician, please, the clinicians will say us to do this thing, then we'll do it in clad only. We'll tell them clad is better media. Okay, that's all. So this is a basic constituents. Now let's go to the types of culture media, which is very important. So this picture you guys must not be having in your notes, so don't worry. You know, we will have it in tomorrow's class. Many places this picture is coming. This is just a brief I show you, and you will be given this file also for you. This PDF you will get, everybody will get. Only few pictures I added, but this picture you have in tomorrow's class. You'll see. Now plus see it, because we are going to talk about the types of media now. So types of media, when you see, this is this I'm only telling. This is a blood agar, how it looks. This is a chocolate agar, how it looks. This is your LJ media for TB, and this is your LSS media, and this is your Robertson cooked meat. This is McConkey, and okay, it's all fine.